All right, welcome to today's extension activity. So when you're in a seated lab, we tend to do these experiments where we're going to test food and different items for the macromolecules that we're interested in. Specifically, we're going to talk about glucose or simple sugars, starches or proteins. Today, I am going to do that for you, and you're going to make your own hypotheses based on your understanding of foods that are in your environment, as well as the positive and negative controls for the tests. So in order to test food items, like if I wanted to know, does my protein powder actually have protein, I have to know what a positive and negative control test looks like. So we are going to be checking for simple sugars using something called a Benedict's solution. Color change will indicate the sugar amounts. We're going to be testing for starches using iodine. Color change will indicate the presence or absence of starch. And then we're going to be checking for proteins using Biuret reagent. Again, color change indicating the presence or absence of that. But to know what that color change is, we are going to test some known substances. We're going to start with distilled water, which should be a negative control for all of our tests. Distilled water should not have any sugars, starches, or proteins in it. We're also going to check glucose. We're going to check starch. And we're going to check something called albumin. Albumin is a protein that we're going to see in a lot of different foods. So we should have then a positive control for glucose with the simple sugar test, a positive control for starch with the iodine test, and a positive control for albumin with the protein test, and then be able to see what negative responses look like for all of these if it's not going to have that main macromolecule in it. So I'm going to start getting this set up, and you're going to make your hypotheses about what these should be positive or negative. All right, so I have added about 2 milliliters of solution to each of our test tubes, and I did that 3 times over because we're going to be running 3 different tests. For our first test, we're going to run what's called the Benedict's solution test, and this should turn colors in the presence of glucose once we heat up the solution. So we might get a really strong response to begin with like pure glucose, but generally we have to heat up the solution. So I am going to add just enough to make it a blue color, and this is distilled water. It should stay that blue color, and I'm going to put it into a warm water bath. And then I have my glucose, and again, no reaction until I warm it up, so that's not overly surprising. I'm going to add my starch, no reaction, and my albumin. We're going to add just a little bit more to the albumin. And again, no reaction. It should be a nice light blue color. And we're going to let those solutions sit in that warm water bath for about 5 minutes before we see the results. But while we're waiting for those results, we can do the starch test, the iodine test, and it should be an immediate reaction. So once I add the iodine to these test tubes, we should see a very quick, immediate reaction. So my iodine in my dropper, I have my distilled water, and no reaction. It stays that kind of tea color, that kind of off yellow brown, which is expected because distilled water shouldn't have anything in it. Glucose is a simple sugar. It is still a carbohydrate, but we should not see a response with glucose. Good, no response for glucose. However, we are testing a starch solution. If I don't see a strong result with this one, someone didn't make the starch solution properly. So let's go ahead and add some in there, and we should immediately see it turn a nice dark color. We get that kind of almost black coloration, and that tells us that there is absolutely starch present in this solution. So that's what we would be looking for a positive starch test. And let's just make sure that our albumin doesn't react. It shouldn't, because albumin is a pure protein. And so again, that yellow kind of color. So we are seeing here 3 negatives and a positive test result, which is expected. Starch should be our positive test result and should tell us then if I test an unknown food item and it turns a dark color in the presence of iodine, we have starch present. The last test we're going to run is the Biuret reagent. This tests for the presence or absence of protein. So we should see no color change if there isn't any protein present, but if there is protein present, we're gonna see it turn a slight pink or purple color. So we're gonna add some to our distilled water. Again, I would expect no color change. No color change. It's not going to even look like there's much color to it because the Biuret reagent is a very pale blue. It's got some copper in it. And again, no response with glucose. Perfect. Should stay kind of that clearish color. If you put it on like a white or a black background, you might see a faint blue color. Starch, no reaction. Fantastic. That means we made all of these correctly. And then for my albumin, I should get a color change. And so we should be seeing kind of a light purple color, maybe kind of a pinky lavender color there. If I added a little bit more solution, I might see a slightly stronger response. So sometimes we add some more solution to get a stronger response because it's gonna bind to those peptides, and that's gonna show us a positive result for protein. So at this point, we have now done our positive and negative controls for our starch and our proteins, and it looks like we're getting some color results on our sugars. So if I pull out my sugars, I'm gonna do them in reverse order. Here is my albumin, still that blue color. That's great. I don't expect there to be simple sugars in a protein. In my starch, again, that's great. No color change, no simple sugars in a starch. In my glucose, I am going to expect a color change. And in this case, we have this bright orange color. So the more orange it gets, the stronger the sugars that are gonna be in it. And so my glucose should have a positive response for a glucose test. And then water, once again, we're having a negative response on that distilled water. So that just means that all of our positive and negative controls happened appropriately. And now you should be prepared to go to the next part and try and figure out what we might see in some of these food items, including our unknown solution. All right, so now we are going to test some food items that you probably have in your kitchen at home. So we are going to use whole milk. So you should write a hypothesis for what you think is going to be in whole milk. We are going to use egg white. So we have an egg, just to show you an egg, and we have egg white. We're going to see what's in that. I also have a banana. So this is a very ripe, overripe banana in my opinion. I like them green, but we're gonna see what's in the banana itself. And then I have a rice solution. So I just have rice that's been soaking all day. So we're gonna see what we think we find in rice. And then last but not least, I have an unknown solution here. And I'm not telling you what's in this. You get to just kind of take your guesses based on maybe the color. It smells a little fruity and a little chicken-y. So there is your hint on that. You're going to pause the video while I get this set up, and you are going to write a hypothesis for each of these, for each of the solutions we're testing. So you should say, I think egg white is going to have blank of these. So maybe you think egg white is going to have starch for some reason. So you'd think that egg white would have a positive result for starch and maybe a negative result for glucose. You're going to write that down in the hypothesis section of your table, and then you're going to come back and we're going to see what those results actually are. Okay? Go write your hypotheses now. All right, so we're gonna do the same thing as we did with our positive and negative controls. I'm gonna set up my Benedict solution. So I'm gonna put a dropper full of Benedict solution in each of our test solutions and stick those in our warm water bath. The warm water is required to get a solid result on this particular test. 4, and then our unknown. 5, and we're gonna let those sit while we run the other tests, which will have immediate results like you saw in the positive and negative controls. So here is iodine with our milk. I am not telling you what the result is. You are going to interpret the result and you're going to write it down in the iodine test for each of these solutions and base it on your hypothesis. So, iodine in our milk. Okay, there is our result for milk and iodine. Iodine in our banana. So there's our result for banana. Iodine in, albumin, that was albumin. Iodine in the banana now. So there's the banana. And we should, there we go. I thought that looked a little funky. So it was milk, albumin, egg white, which is albumin, banana, iodine in our rice water. Okay, so rice water, and then iodine in our unknown. Let's see if there's any starch in our unknown. All right, so there are our color results for our iodine test. Again, here we got those right up there, and they're in that order. So milk, egg white, banana, rice water, and unknown. Okay, hopefully you got those good results. And we're going to do the same thing for our protein test using the Biuret reagent. So for our protein test, we have our milk, Biuret. Let me do 2 drops here because Biuret is very faint. And we might see a color change there. I hope that's showing up on the camera. It's light purple, so I'll let you guys interpret that. So milk turned a light purple. Here is our egg white. Okay, we should get a result there. Banana. I don't know if we'll see protein in banana, but let's find out, test that hypothesis. Okay. Rice water. Let's see what we get for rice water here. Okay. And then last but not least, our unknown. Let's see what we get with the unknown. Now, sometimes we have to remember when we're testing foods, we have some colors involved in those foods, which may or may not affect our results. I'm not seeing a lot of color there, so that looks like it's just the same color as, again, our unknown is kind of this brownish yellow color. Put a little more in there. Nope, it looks like it's staying the same. So do that what you will with your results. And we're gonna let this water run a little bit longer because it is currently not really giving good results. Sometimes it can take 5 to 10 minutes. So come back and I will get those results for you. All right, so we let our Benedict's solution run for another 5 minutes in the warm water bath. And with Benedict's solution, we can get a range of colors as a positive result, where if we see anything that's kind of a yellow-green all the way up to a really bright red, so like yellow-green to orange to bright red, can tell us the presence of sugars. And it can indicate that there's more of those sugars present with the brighter color response we see. You also have to consider that we are adding this to some colored foods, and so we might see some results that we have to interpret based on the color of the food itself. So with that being said, here are the results. We have our milk, okay? We have our albumin. We have our banana. We have our rice water. And last but not least, we have our unknown, which, to remind you, the unknown does have a faint beige color. So we have our unknown there. Hopefully you were right with your hypotheses, guessing which macromolecules were in these food items. Record all of your results in your lab notebook, answer those extension questions, and get everything submitted with this week's lab. Thank you.